Our next speaker is Veronika Yumiseba. She's from Lübeck in Germany. She's a Hurley Career Awardee candidate, and she will speak about next generation functional genomics, single-cell, CRISPR-optimized approach for uncovering target genes of coronary artery disease variants. Veronika.
Hello, everyone. I would like to thank the organizers for allowing me the opportunity to present today in front of you the work of my PhD thesis. I have nothing to disclose in this presentation, so let's start. Nowadays, we can say confidently that we are in the next generation genotyping era. Most of the patients that come to the clinic get their genome sequenced. So now sequencing is not a challenge that we are facing. The biggest challenge that we now have is that we find mutations all over the genome. Some of them are in the coding genome, and their impact is slightly straightforward to decipher. But most of the mutations found are in the non-coding genome, and their impact is not so straightforward, and it demands functional validation. So if one would like to do functional validations on them, one would create a mouse model. But as I said before, if we look into the entire genome, we end up having many of them. It would mean many mice to be created. But realistically, this is a very labor and very time-consuming project, so realistically, few of them could be created. So here we have a bottleneck where we have many more variants than the ones that we can actually test. So what do we need now? We need a next-generational functional testing for non-coding variants. So which diseases are the source of non-coding variants? The answer will be complex traits. So today I would like to introduce to you coronary artery diseases, which are complex traits, and through GWAS, very large ones and ethnically diverse, several single nucleotide variants have been associated already with this disease. So far, 393 loci have been associated to coronary artery diseases. 95% of the variants found in coronary artery diseases are sitting in the non-coding genome, and 60% of them are in potential cis-regulatory elements. It means that here we have a great potential of fishing enhancers or regulatory regions. Again, we face the same problem, that we need to create one assay to test all of these single nucleotide variants. I'm bringing to you today this Manhattan plot that helps us interpret GWAS results, and I just would like to highlight the single nucleotide variant that is here, having the very small p-value, and this is named as the lead variant. And one of the problems while identifying the lead variant is that some of the cases, this is not the functional variant. Why this is not the functional variant could be that the actual functional variant did not reach a significance because of frequencies in the population analyzed, and also due to the methodology that GWAS used, which is array. So it means that not every single nucleotide variant is being sequenced. So if one would like to investigate which will be the causal variant in this particular region, one should consider not only the lead variant, but also other variants that are in the surrounding. There is a dogma in GWAS that the single nucleotide variant affect directly the genes that are in the neighborhood. So how can we test if these single nucleotide variants are sitting in a cis-regulatory element? So I will introduce you today to CRISPR technology. And for doing this, we need first to have the guide RNA, which is bringing the machinery to the particular location that we want to study it. In this case, I would like to introduce you today to the dead Cas9. So this is a Cas9 which is not actually cutting the genomic DNA, but is bringing attached with it some domains that could be either activating domains or interfering domains. In the case of using CRISPR activation, if we are truly sitting in an enhancer, then the genes that are directly affected by these enhancers will upregulate it. But in the mirroring technology in CRISPR interference, then we will see that the targeted genes will reduce its expression. So what is the goal that I am now going to present that was the motivation of my PhD? We wanted to generate next-generational functional genomic assay to test hundreds of loci in just one single experiment. What we used, first we used CRISPR screens, activation and interference, and also we will use single-cell RNA sequencing as a readout. So how CRISPR screens work? First, one have to have their targeted cell. In the case that I'm presenting today, I've worked with monocytes and endothelial cells because they are crucial in coronary artery diseases, and I make them express the dead-Cas9 plus all the machinery that I need. And in the case of a CRISPR screen, one is delivering not only one guide RNA at a time, but a complete and very complex library. For this case, we use lentivirus as a vector to deliver these to the cells to make the system more efficient. So it means that after combining the cells with the guide RNAs, we will have in a Petri dish cells expressing one guide RNA, but also other cells expressing more of them. We will use single-cell RNA sequencing because with this technology we could identify which are the transcriptional changes that one guide RNA or several guide RNAs are driving. So let me talk to you about the guide RNA library that we use so far. We have targeted 220 non-coding loci associated with coronary artery diseases, and as you can see here in the circus plot, almost all the chromosomes are represented. We also use positive controls, which are guide RNAs targeting some transcriptional start sites of specific genes. And we also use negative controls, which are guide RNAs targeting regions in the genome that do not have genes in the surrounding, and also random regions, which are regions that are not present in the human genome. In total, we have a library of 2,000 guide RNAs, and we tested them in 300,000 cells. It means that we perform 2,000 experiments in just one single dish. For the data analysis, we've used Cell Ranger to create the matrices, the cell gene matrices, and also the cell and guide RNA matrices. For the single-cell RNA sequencing analysis, we use Seurat to do the QC and then the further exploration of the data. And then we use a tool that is called Scepter for doing the differential expression analysis. All the pipelines that we've created throughout the thesis, they are in a repository in GitHub. So let me share with you the results that we've got, and due to time restriction, I will show it to you just one locus in the two cell lines that I've been working with. And first here in endothelial cells, I wanted to show it to you that every single dot here represents a single nucleotide variant that we've targeted with the guide RNAs. And upon interference, here, for example, we detected that SMAD3 was downregulated. And I just want to point out that every single dot here in this box plot represents a cell containing a specific guide RNA targeting this particular single nucleotide variant. There were also other genes where its transcriptional changes have been discovered once we did CRISPR interference. One of these was PS1. And using the same cells which are having the guide RNA, we detected also transcriptional changes in a different gene within the locus. In the same cells, we performed the activation experiment, and we also detected that SMAD3 was significantly upregulated, and this change was driven when the cells contain two different guide RNAs. In the case of the monocytes, this is exactly the same locus that I'm showing to you. And at the very first spot or site, you can see that there are cellular specific changes. And once we did the CRISPR interference, PL3 was downregulated. And when we performed the activation experiment, that were SMAD3 and SMAD6 were significantly upregulated in the presence of the same guide RNA. Just to sum up the results, when we perform CRISPR activation in endothelial cells, 19% of all the guide RNAs show positive results, and in monocyte, 12% of them show positive results. When we perform interference, 23% of all the guide RNAs show a positive result, and in monocyte, we identify 5% of all the guide RNAs. I would like to bring this figure again to the presentation, and just to tell you or remind you that the dogma that GWAS has is that the single nucleotide variants affect directly the nearest gene. which we saw that this is not always true, because in a higher percentage of the gene expression changes happen not only in the gene that is just next to it, but in other genes in the locus. We wanted to do one step forward, and we wanted to validate these newly discovered cis-regulatory elements. And we use now the CRISPR machinery, but we use now the active Cas9 because I wanted to remove this newly discovered cis-regulatory element from the genome. Then I pick clones to just make sure to pick clones which have an homozygous deletion of this cis-regulatory element, and then I perform RNA sequencing in bulk. I just want to bring again this result from the single-cell RNA sequencing, where I told you that SMAD3 was significantly upregulated, and I'm glad to say that also in the validation experiment we could replicate the same effect. I told you that upon interference experiments, RPL4 was downregulated. We could also saw it once we performed the validation experiments. I just want to mention the advantages, but also the limitations that we have while we are performing this CRISPR screen at a high throughput level. Among the advantages, many loci could be tested at once. We could identify cell-specific changes. We can test not only our favorite gene, but many genes in one experiment. We could identify cis-regulatory elements, and not only that, but also the genes that these cis-regulatory elements are affecting. We have performed our experiments in the genome context. Among the limitations, we do not need to forget that there could be off-target effects of the guide RNAs, that we're delivering a very complex library to the cells, and due to this reason, some guide RNAs could drop out. It's important to bear in mind that the higher the variant the effect has, the higher the chances of the effect to be picked up. And obviously, we're collecting data which is very complex, and it implies some bioinformatic skills to analyze it. I would like to apply this approach perhaps to other coronary artery disease relevant cells and validate even more our results, because we know that enhancers are cell-specific. I would like to also use this approach for other diseases, and in our interest in our lab, we wanted to apply this technology to validate these de novo non-coding variants discovered in patients. As a conclusion, I hope that I convince you that using this approach, one can identify cis-regulatory elements, in this case in coronary artery diseases, and not only that, but also the genes that are directly affected by these cis-regulatory elements. I want to finish the presentation, but I would like to thank for all the support to my supervisor, Dr. Malte Spielmann, and also all the lab for the great collaboration. Thank you very much, and I'm happy to get questions.
Questions for Veronica? Yes, announce yourself, please.
Hi, David Garfield from Genentech. Very nice work. And my question is about the way in which you're perturbing the variant, right? So you're perturbing the variants in this case with what is probably fairly large, right? So the CRISPR activation or repression domain is going to cover the whole enhancer, whereas SNPs are going to be like one position. And so I was wondering if you'd looked at all into either an EQTL baseline or how this kind of perturbation compares to prime editing or something where you're looking at just one variant and not necessarily shutting down the whole CRE.
Yeah, so for the design of the guide RNAs, I know that the single nucleotide variants occupies a position, so we look at 200 base pairs in the surrounding, and then we design the guide RNAs. One has to bear in mind that for this particular approach, I use the Cas9 that needs an NGG, so a PAM sequence, so it means that I have to look within these 200 base pairs, a PAM sequence, and then for the guide RNA design. And then, obviously, every single locus, I do not use, as I say, the lead SNP, but also SNPs in the surrounding. So it means that every single locus was represented with 9 single nucleotide variants. That's why the library was huge.
Yes, go ahead.
Yes. Hello. Henrike Heiner from HPI, Potsdam, Germany. I was, very cool talk. So I've been sort of similarly as the previous question, wondering if you've been thinking about comparing this with expression QTL data from GTAC also.
Yeah, definitely. This data is recently generated, so we just analyzed the data, but of course we wanted to do some bioinformatic analysis to compare if there are other databases, as you say, for example, this EQTL data, and if this correlates to our data. That's the next step that we are going to do, yes, of course.
Thank you, Veronica.
And this brings us to the third speaker of this session.